today is about preparing us for a tiny piece of work. A tiny piece of work from your grade 11-12 piece of biology that we need to cover. But this covering and writing a test is not about, I want to take a short cut and I want to get it over and done with. It is about testing yourself. It is about slowly starting to realize the end is closer than what I think. And it is about getting myself ready. So, yes, I am going to show you a few short, important information pieces to help you for this test. But actually what I want to do is I want to invest another bit of information so that you can be prepared for the end of the year. You are going to write a test on the pages that I indicated. It will be on what is DNA, where do I find DNA. It will be how DNA can synthesize itself, and then also how proteins are being synthesized. I see a chromosome. And a chromosome is a length of DNA with a lot of genetic information on it. That length of DNA is actually the material that is called up. And if I can get to the smallest piece of DNA, I see DNA is a double helix, and I can see that this double helix consists out of a backbone with a pentose sugar and a phosphate group, which makes the backbone. But in between, I have 4 different types of nitrogenous bases: adenine, thymine, cytosine, and guanine. But they link with a complementary pair. So G always to C. And what I also see is that G and A are the larger ones. T for that, A and G are 2 rings. And we call them the purines. And then we have the pyrimidines. Thymine and cytosine are the pyrimidine. And we see that they bond with hydrogen bonds. And there are 3 hydrogen bonds between guanine and cytosine, and 2 hydrogen bonds between thymine and adenine. Okay, and if we need to get to the other type of nucleic acid, we have RNA. We also know that RNA is a bit different from DNA. The first difference is single strand, double strand. Because this is a single strand, no complementary strings, therefore no hydrogen bonds. Then I also see that there is a uracil and there is a thymine. Right. That is important to remember. I see that the sugars differ. Ribose sugar, deoxyribose sugar. The difference is this large DNA cannot leave the nucleus. This one is small enough to leave the nucleus. When We have the replication of DNA. That is for the cell to divide and make two identical copies of the cell. When do I want cells to divide and make identical copies? I want it when I want growth to take place. From zygote to embryo, I need an increase in the amount of cells. Mitosis, when I need to replace worn out cells, or when I'm cut and I need to heal, there need to be exactly the same cells. So that is when I need an identical copy. What will happen? The DNA will unwind, unzip. Helicase will be the enzyme for unzipping. Remember, there's a mistake in the textbook. So unzipping. Then the loose nucleotides. Nucleotides? Yes, a nucleotide, I know, consists out of a phosphate group, a sugar, and a nitrogenous base, which will attach and form a complementary string. I will have the original string, which serves as a template. And the DNA polymerase will build up this formation so that I have again two DNA. The outer ones was complements of one another. Therefore, the one that links up there, these two green ones will be the same. Now we say we have semi-conservative replication. Why is that a good thing? Because I always have my true old template, and the nucleotides that's attaching has the correct sequence. Why is sequence important? Because the sequence of nucleotides eventually determine the sequence for my protein synthesis. And then, of course, protein synthesis. And I dare you, grade 12, see if you can fill in. See if you can fill in. Let's first fill in what is the name of this process. What is the name of this process? This process where the DNA comes and get the code. So without wasting too much time, transcription, get the copy. Get the copy but in a complementary form. And now moving out into the cytoplasm, and now we have translation where the anticodon attached to the codon. Accommodation is the ability of the eye to focus on distant and near objects.
And I bring it in contact with the electroscope. What you must keep in mind is positive charge can't move. It is the negative charge that moves.
So the first one, EF, is then going to be 0 minus negative 2 over 4 minus negative 4. Look at my signs. If you go wrong there, if you don't substitute in brackets, you're definitely going to get wrong answers.
It's fun time, it's play time, so please play along. Revision means I am now going to see what do I know and apply that. And then if you realize, but something is not correct, see if you can correct it, or please contact me and ask me why is how you think perhaps not correct. Okay, so let's start with inheritance, and we are going to revise all the possible inheritance theories that we did study. We are going to look at a few of the terms that are important. Dominance and recessive alleles in a gene, codominance alleles, multiple alleles, the sex-linked problems, and then what is the gender of a baby. That is a short revision so that you just can see whether you do follow the work or not. The terms that I want you to look at is haploid and diploid. A nucleus is haploid or diploid. That means that in a nucleus, there can be a single set of chromosomes, meaning in the nucleus there is only one of the pair that is in the parent, one single set unpaired chromosomes, and that forms the gametes. But in a diploid nucleus, there is two sets. That means both from the father and from the father and from the mother. From the father and from the mother. There is from both of the gametes fused together, and now we call that a homologous pair. Now you see a homologous pair. So, if there is a nucleus that have only one of each of the homologous pairs, we will immediately know that cell is a gamete. We will recognize a gamete that the chromosomes are single, there's no homologous pairs, there's only one set of the chromosomes, haploid. And we refer to a haploid nucleus as N number of chromosomes. That N will mean in human cells 23. You do follow that. A specific fly has 6 chromosomes. Yes, that small amount. And that means N means 3 for that fly. Diploid means 46 in human terms. Good, let's continue. Then we have our chromosome homologous pair. With this we mean that these two are similar. And I want to use that word similar. These are, sorry, let me just get. These two are similar because they have the genes for the same characteristics. It is not the same genes. We can see the gene that is on that locus is the gene for a specific characteristic, but it is not the same. No, they will be the same size, and they will be on the same location on the DNA. They will have the gene for the same characteristics. They will be the same size and they will have the same location on the DNA. In that way, they will be similar, but they are not necessarily identical. Of course, if it is an identical twin, they will be the same. But, no, sorry, not even then they will be the same. They will be the same in the two individuals, but not the same there. These two are not identical. Why can they not be identical? Because a gene has a specific locus, or genes have loci on the chromosome, and the two alleles can either be the same, which means the same allele has been inherited, or it can be different. If it is the same, we say this individual is homozygous for this characteristic, but it will be heterozygous for the characteristic that will play out in A. This is homozygous for the dominant feature, and here we see homozygous for the recessive feature. Good. I hope that you do follow this. Now, homozygous, dominant. Why do we have different colors? Because one is inherited from the father and one from the mother. Then, homozygous dominant, yellow, is the dominant allele. This gene, let's say it is for eye color. Both parents had the dominant allele in their gametes, or the recessive allele. Or if you can inherit the dominant allele from one parent and the recessive allele from the other parent, that is a heterozygous genotype or phenotype. Good. So, let's take a problem. 2 black mice, one heterozygous, the other one homozygous for coat color. 2 black mice. Recessive allele is gray. What possible colors for the offspring? 2 black. So, I will start with the parental phenotype. It is black that crosses with black. The one is heterozygous, and the other one is homozygous. So, the heterozygous will be capital B, small b. And I normally will write down black is dominant, ne? And what will grey then be? The grey coat will then be small b. Important. We use the small letter of the dominant allele. And the other one is homozygous. Now there will be gametes. Immediately I draw circles and capital B, small b. So, if this is the male, this will become sperm. And the sperm can either have a B, a capital B on it, or a small b, because to form gametes, meiosis take place. The homologous pair separates, yes, so there can only be one. Now I have a haploid cell. And there we have that possibility. Now show fertilization, please. And I am going to use different colors to show fertilization. So, fertilization can happen between, there's a possibility that it can happen between those two, or between those two, or this one and this one, or this one and this one. This is all possibilities. Now, each time when there is intercourse, this is the possibility that can take place. So, we can have the genotypes will be B, capital B, small b, or that one, capital B, small b. So, the genotype will be, there's a homologous black, and there will be one. And then there are heterozygous black and 3. All of the offspring will be black. But phenotypically, all will be black. But when we look at the genotypes, there can be a homozygous black and a or heterozygous black. And now, grade 12, it doesn't matter what is the problem. Whatever they ask, you write it down like this. If they ask a genetic diagram, you have to use all of this in your answer to have full marks. They can also ask the question many different ways. Sometimes you have to do a bit of calculations, but you are higher level. So therefore, you must think. So therefore we have another problem. 2 black mice, 8 babies, yes, 8 babies, and 1 is grey. What is the genotype of the parents? Show it with a test cross. This is where I do not know the parents. I can do a test cross. So, if I have a baby and the baby is grey, again, we have black as dominant, and the baby is grey, so the little baby will look like this. I write in here, small b, small b. This is telling me that from this parent, this baby inherits a small b, and from this parent, the baby inherits a small b. You agree? That is how I will start. If there is one grey baby, there is the possibility that, not the possibility, there must be a B, a grey B, and a grey B from both parents. If I look at 8 babies and 1 is grey, it means 7 is black. So black, 2 grey, more black ones, and the parents are black. So the parents must be heterozygous. black. That gives me 1 out of 4 will be grey, and the rest will be black. But if I look at that, that is not the perfect ratio. But it is telling me that the smallest amount is grey. So therefore, the parents was heterozygous black, phenotypically. Their genotypes was capital B, small b, both. That is the only way how the offspring can turn out like that. Okay, now a Cftr gene codes for a specific transport protein found in cell membranes. Cystic fibrosis is an inherited illness where a mutation in the Cftr gene causes chloride ions to build up inside cells. When chloride build up, it will change the osmotic potential. It will have a lower osmotic potential, and the water will move into that cell, which causes them to absorb more water by osmosis. Very important. In the lungs and pancreas, this occurrence leads to the production of thick, sticky mucus that can affect breathing and the production of digestive enzymes. From the pedigree diagram, what can you say about how cystic fibrosis is inherited? Explain your answer. So I have female and male, and the colored ones are the affected ones. If I look at the parents and the F1, the first generation, I again see one has cystic fibrosis, the other two does not have cystic fibrosis. So this offspring did inherit a cystic fibrosis, and here they tell me I must use CF. So this individual, number 2, did inherit the genes. So I will say cystic fibrosis is a recessive allele. My answer will be 1 out of 3 offspring contains the disease, and even that the parents are both not affected, they do have an offspring. So 1 out of 3 is my explanation of the offspring, and the parents, although they are unaffected, did produce an offspring that has cystic fibrosis. Therefore, it is a recessive allele. Do you realize that this, I just want to show you, this is the offspring. Now, here we have another marriage. Here we have another couple. Can you see that? Important to see. That is important to understand. Here we have a marriage and the offspring. There we have the marriage, and that shows the offspring. Okay. That is important to understand in a pedigree diagram. Write out possible genotypes for individuals 1 and 2. So, because there is a small cf, both must contain a small cf. Here they use the cf. We normally use only 1, but I think there it's capital small, because that one did inherit a small. Now, What is the probability that individual in line 2, number 3, is heterozygous? Is there a possibility? I think the possibility is 100%. Because, no, sorry, it is not 100%. I don't agree with myself. What is the probability? The probability is heterozygous, heterozygous. When we test heterozygous with heterozygous, we know that the possibility is 1. I'm going back to this one. Heterozygous, heterozygous always gives me 1 dominant there is 1 dominant, 2 heterozygous, and 1 recessive homozygous. Nah. 1 dominant homozygous. So, if I go back to that one, we have number 3 there. The parents are heterozygous, so the possibility to be heterozygous is 50%. To be heterozygous. Just look, what do they ask? What is the ratio? Then you had to say 1, 2, 2, 2, 3, 4. Homozygous dominant, heterozygous, homozygous recessive. Now, then they ask the ratio. We can say 50%, or the chance is 1, 2 out of 4. That is 1/2. Now, you can also use it as a fraction. Read carefully, what do they ask? What is the possible genotype of individual, and number, and let's say, I think I wanted to ask number 5. What is the possible genotype? This one we know is small c, small c. This one, capital C most likely. Why do I think that? Because no one here has cystic fibrosis. This one can look like, will definitely look like this. Even if that one was small c, capital C, because it's unaffected, so it must have the dominant gene. Now, to mask off the recessive one. But this parent has a small c, so this must be a heterozygous. So we call those that is looking like this, we say they are the carriers. Now, heterozygous, where a disease is involved, are carriers. So this number 5 will be a carrier. Okay, right. Let's go to codominance. Codominance, both alleles shows dominance, exert their features. If we look at the heterozygous, the parental phenotype of our fish there is red and blue. But when I see the offspring exert both of the features, there's red and blue, I know I must use a C. Now, and I use a C, R, because that one is red, and a C, B, B for the blue ones. So, the moment when my gametes form, it will look like this. When fertilization takes place, I can have a CR, CB. You agree? And that is the only combination that I can have. So I have an individual that shows both the colors or both the features, and therefore it is CRCB, and the C means codominant. So we have a blue-red or red-blue individual. Of course, when two of these will mate, there will definitely be red ones and blue ones, and of course some blue-reds, or red-blues. That is then the possibilities. Okay, grade 12. Now I want us to look at multiple alleles. Multiple alleles, you know blood groups. Yes. And blood groups, we have. Blood groups, we have phenotype. Can we write down all the blood groups? We have A, B, AB, and O blood groups. The genotype for blood groups, we use an I. The I is for intermediate, but don't break your neck or your bones. Just remember, C for codominance, but blood groups is an I. Okay, so if I am blood group A, I can be homozygous or heterozygous. If I'm blood group B, I can be homozygous or I can be heterozygous. Blood group A, B, there are only 2 spots, remember? But we have 1, 2, 3 different alleles. We have 3 different alleles. Why? Because A and B is codominant. So I can have A and B together, and that makes it possible because they are codominant. But O is recessive. And therefore, if I am blood group A, I will be A because A is dominant, so homozygous, or A and A is dominant, and therefore this will also be blood group A phenotypically, because the dominant one will exert its features. Okay? That is very important to understand. Let's go then back to our problem. What is the possible outcome of an heterozygous mother? And she's A, so she will look like this, heterozygous. I must remember A, she will be blood group A because O is recessive. And a homozygous B father. Homozygous B father. And the offspring phenotype will be AB, phenotype, B, phenotype, AB, phenotype, the phenotypes. What is the blood group? AB, B, AB, B. So it's a 1-to-1 ratio for AB to B. That is the phenotype. Genotype, IAIB, IBIO. That is the genotype. Good. Now, can you read this or can you not read this? I hope that you all can read this. I see 7, 13, 16, 9, 12, and an 8. If you had trouble to see any of these letters, it is because maybe you are colorblind. That means that the message from your optic nerve that is taking it from the rhodopsin and the iodopsin, from the rods and the cones, especially the cones, colorblind cones, to your brain is not working properly. And this is a sex-linked disease. Why? Because when we look at the gonosomes or the sex chromosomes, we have one pair of sex chromosomes. Yes? That one pair consists out of an X and a Y if you are male. The Y do not have that space to have always the other allow to mask it off. So, therefore, the male can have a disease and it cannot be masked off. The gene is carried on the X. So, color blindness, mother give it to her son. No, I do not want you to use that word give. The son inherit the condition from his mother. And let's quickly do a Punnett square. For a son to inherit it, it means it is carried on the X. A mother is not colorblind. She is not colorblind. It is this recessive gene, this tiny one, that will cause colorblindness. The father is not colorblind. The father is not colorblind. Right. But there is a Y. There's not an X, so there's no C on it. So, this will be a girl. This girl, capital C, small c. This girl, capital C, capital C. Okay. Just to make sure you do see it properly. This is the colorblind recessive small c. There it is. Now, the father, not colorblind. This will be girls. Very important. This will be girls. These girls are not colorblind, although the mom can be called a carrier because it is a condition. She's a carrier. One possibility for her girls, if the pregnancy ends up to be a girl, there is a possibility that 50% chance that she is a carrier. The boy will look like this. X from its mom. X from its mom. Y from the dad. But on the Y, there is no allow for colorblindness or not colorblindness. There's nothing. There, this child will inherit a small c. And this one, so 50% chance that this mother and this father may have a color-blind son, 50%. That means 1 out of 2, that if this is the mother, she's a carrier. How will she know she's a carrier? If her father was colorblind. You agree? She can be a carrier. Interesting. Another problem is hemophilia, which is a blood disorder in which certain blood clotting factors are not produced. This results in excessive bleeding that can damage organs and tissues. Hemophilia is also an X-link, and it is a gene mutation. It is more often seen in men than women, and this is the same reason. If you are female, to have the disease, you need to be like this. You can be a carrier. Capital H means not hemophilia. You agree? Because the X chromosomes are large enough to have the allele, 2 alleles. But if we look at male, remember it's only carried on the X. So either you are colorblind, you have not hemophilia, but if your mother had hemophilia or she is a carrier, there's a 50-50 chance that you will inherit it. Right. I hope you do understand that one. Then, of course, is it a boy? Is it a girl? So I want you first to understand that our gametes, yes, the gametes can either be X can either has an X, a gamete can has an X, yes, or an X, if it is a female. But remember, that is not the only chromosomes. No, there are 22 other ones. So if I have to state a gamete, I will write it like that. If I look at the male gamete, it will look like this. X plus 22 or Y plus 22. That is how a gamete look if I write down all the chromosomes. But when I want to show a body cell, I will say, if it's a female cell, it will be XX plus 44. You agree? And if it is a male body cell, it will be XY plus 44. I hope you understand that one. So I just want to erase this. You see I'm doing revision, nah? Good. So this will be female gametes, and the male gamete will look like this. So will I have a boy or a girl? With each pregnancy, what is the possibility? The female will have X or X. female for gender determination, only an X. The male, a X and a Y. So who is going to determine the gender of a baby? The male. Because the female has only an X. No. She can only add an X to the baby. It's only the male that can decide. If the sperm that do fertilize the egg, that does fertilize the egg, is an X, it's a girl. If the sperm that does fertilize this egg is a Y, then it is a boy. But there's always a 50-50%. One chance to have a girl, one chance to have a boy. According to this. This is called sex determination.
Book Kushuna kuvuso ndikirikiki. Hey, yes, I'm so, there's no man who can yatula. Yes, I'm so, there's no man who can swallow. Okundivona ndikirikiki. Kushuna kuvuso ndikirikiti. No rulimi wembili mbili. Kuwamba hawa mbulingilingi.
Okay, we my niggas, what we starting now. It's so cold on the tempo, you know, ya pita. It's a black car, na mama young. I'm sorry if I did it and you never like it. My God is a blessing, and we gonna make it far. When you hitting on us, you can never beat us. It's sorry, it's sorry, it's sorry. If I had a feelings, oh, sorry, sorry, sorry. I'm sorry.
This is the Football Review. Gareth Bale was brilliant for Real Madrid, worth all of the 100 million euros the club paid for him. If you talk to me about what it means to finish second rather than third or fourth, I think second place is still first among the losers. It might be suggested Chelsea is still counting the cost. After scoring 107 goals on the way to the 2017 Ligue 1 title, Monaco managed just 40 from 28 games when last season was called off. History is written by the winners. That is what is remembered. The three most expensive transfers in Barcelona history are shaping as disasters. Injured more often than not, Ousmane Dembele and on loan Philippe Coutinho have had minimal impact. Struggling Antoine Griezmann has hardly been worse.